Rough endoplasmic reticulum has also with it the secretory, which is the Golgi apparatus. So, well-developed rough endoplasmic reticulum and the well-developed Golgi apparatus, a lot of free ribosomes, and as well as many mitochondria. So we have many mitochondria, we have rough endoplasmic reticulum, we have Golgi apparatus, and we have a lot of the free ribosomes. As regards the nucleus, it is achromatic nucleus because it is an active cell with the prominent nucleolus. Please remember these features because these are the features of the protein synthesizing cell. Again, they are the well-developed rough and Golgi, numerous mitochondria, numerous ribosomes, achromatic nucleus with prominent nucleolus. Then, what are the functions of this cell? Here, we will move to the function. The functions here, of course, it is protein-forming cell, so you will expect to have the following functions. Number one, the synthesis of the collagen fibers, the elastic fibers, the reticular fibers, so the formation of the fibers of the connective tissue. Number two is the secretion of the ground substance, which is present in the matrix, ground substance, which is a part of the matrix of connective tissue, which is a second component of the connective tissue. Also, the production of growth factors that will help the growth and the differentiation of the different connective tissue cells. So this is the third component. Number four, after injury, this cell will help the healing of the connective tissue after the injury, and this always lead to the formation of scar اللي هي الندبة. يبقى إذا هنا the fourth function is the healing of the connective tissue with the formation of the scar أو الندبة. يبقى هنا الفانكشنز بتاعتنا هي لها علاقة بتركيب الكونكتيف تيشو، تكوين الفايبرز اللي هي بروتين in nature، تكوين الجراوند substance which is one of the components of the matrix، production of growth factors that help the growth of the cells and their differentiation, and finally after injury of the connective tissue, by these three functions they can help in healing of the wound in the connective tissue. Let's now move to the other cell, which is the old form of this cell, which is the fibrocyte. This is the fibrocyte, but we should remove these particles here to have more space. As regards the fibrocytes, it is the old form of the fibroblast. As regards the LM picture, you can differentiate between the old form and the fibroblast. How? By the following points. Number one, here by the LM. By the LM, we have fewer processes. This is number one. Number two, the cytoplasm here is less basophilic. Why? Because it is a less active cell, so less rough endoplasmic reticulum and less ribosomes, so it is a less basophilic cytoplasm. And the nucleus, on the contrary, it is dark nucleus. Why? Because lesser activity, so the nucleus will be darkly stained. Look at the picture here. This is the nucleus of the cell. It is characterized by being darkly stained. Actually, the whole processes cannot be seen here. We have just three processes, and this cell, you cannot see the all processes. So the main component to differentiate in tissues or in slides by the shape of the nucleus. because in this slide we cannot see the branching, so we cannot differentiate by the number of the branching. The most sure method is to compare by the shape of the nucleus. Here it is pale with prominent nucleolus, there it is dark and the nucleolus cannot be seen. This is as regards the light microscope. As regards the EM, it's very easy. Here we have the less number of organelles، يبقى هيقلوا الأورجانيلز بتاعتنا اللي احنا قلناهم، in addition to the heterochromatic nucleus. But because it is a less active nucleus in a less active cell. As regards the functions, what are the functions? Functions here, number one, it is responsible for the maintenance, صيانة, of the connective tissue by formation of the extracellular components of this connective tissue. يعني extracellular يعني what is present else the cells أو instead to the cells. We have fibers and we have matrix. So it forms a little amount of the fibers and the matrix for the maintenance of this connective tissue. Number two, in case of injury, this inactive cell rapidly changes to the blast form to help the healing of this connective tissue. This is a very nice picture for the EM of the fibroblast. Look here, we have, this is a diagram for the EM of this cell, which is a highly active cell. You will notice the Golgi apparatus, the rough endoplasmic reticulum, and the diagram showing the formation of the collagen. At first, the collagen is secreted in the form of tropocollagen, and then it will be collagen fibers of type 1 or type 3. This is collagen fibers, and type 3 is the reticular fibers. So it forms the collagen and the reticular. Not only that, but it forms the tropoelastin that will form the elastic fibers. So it forms 3 types of fibers. Not only that, but it forms also the ground substance which is present in the matrix. A look at the actual EM in this left figure. You will see here, first of all, this is a nucleus, euchromatic, and these are multiple or numerous mitochondria. And this is the saccule of the Golgi apparatus, and this is the rough endoplasmic reticulum. So these are prominent in this active cell. And in this picture, you can see the cross-section of the collagen fibers that are secreted by this active cell. This is a cross-section and this is a longitudinal section. Notice in the longitudinal section you can see striations. These striations are called periodicity. And in the next slide I will explain what is the cause of this periodicity. This periodicity or striations only appear, of course, in the longitudinal section, but not evident in the transverse section of the collagen fibers. This picture to explain the periodicity of the collagen fibers. Actually, this is the collagen molecules forming the collagen fibril, and the collagen fibril will form the collagen fiber, and the fibers will form the bundle. If you look here, you will see the collagen molecules having a head and a tail, and the heads are not on the same level in these forming collagen molecules. So we have an area where there is a gap. Here we have 1, 2, 3, 4 collagen molecules, but there we have 1, 2, 3, 4, 5 collagen molecules. This area is called gap zone, and this area is called overlap zone. So we have this periodicity of alternating gap zone, where there is a depression, and the overlap zone. And this is the cause of the periodicity evident in the longitudinal section of the collagen fibril. And this arrangement, of course, makes the collagen fibers are very strong and very difficult to be separated from each other. This is a unilocular white fat cell or adipocyte or adipose cell. It is originating from the UMC, as we know, and the UMC will accumulate fat to form lipoblast, and the lipoblast will give rise either to this unilocular cell or multilocular. As regards the site, it is present in white adipose connective tissue. As regards the LM, it is large oval cell. What is the size of this cell? It is from 50 to 150 micrometers. So it is oval cell, large in size, and it measures from 50 to 150 micrometer. The nucleus is compressed at one side, giving what is called signet ring appearance, where the cytoplasm is full of fat that dissolves during preparation of the hematoxin and eosine sections, giving this appearance of signet ring. As regards the EM picture, we will expect what? Here we have in the EM picture, this cell is full of the smooth endoplasmic reticulum, together with filamentous, some filamentous, not much, some filamentous mitochondria. Fat droplets here appear to be electron dense, so that the droplet here will be electron dense, dark black. This is the EM picture. Lipid droplet 1, dark black or electron dense, and the cytoplasm contains just the smooth endoplasmic reticulum and poorly developed rough endoplasmic reticulum, and as well as few filamentous mitochondria. Few filamentous mitochondria. Don't forget that in the other picture, here the fat can be demonstrated. Why? Because it is stained by Sudan 3 by orange color. So on the right side here, we have the fat cells in hematoxin and eosine section showing signet ring appearance. This is the nucleus, eccentric, compressed, and this is a very thin rim of cytoplasm, and this is the large one droplet present in this cytoplasm. On the left side, we are seeing the fat droplets stained orange with the Sudan 3. It can be also stained by Sudan black by black color. Don't forget the functions of these fat cells, which are the following. These functions are: number 1 is the storage of fat. Number 2, support delicate organs like the kidneys and the blood vessels. Number 3, the heat insulator. Heat insulator. This is a picture for the LM of the multilocular or brown fat cells or adipocytes, and EM for the same cell. The origin of this cell is the same as that of the unilocular, which is the UMC after storage of fat to form lipoblast that will give rise to this brown fat cell. As regards the site, it is present in the brown adipose connective tissue. As regards the LM, actually, number 1, it is smaller than the white fat cell. Number 2, it has multiple fat droplets and not one. Number 3, the nucleus is rounded and not compressed like that of here. It is rounded, not compressed as that of the white. So the nucleus here does not show signet ring appearance, and also